Xenopus laevis egg extracts are useful as cytoplasmic and organelle material for in vitro reconstitution of cellular events, such as nuclear assembly and nuclear protein import. When sperm chromatin DNA is added to either crude or fractionated Xenopus egg extracts, membrane vesicles present in the extract bind to the surface of the chromatin, fuse, and incorporate nuclear pore components to form fully functional synthetic nuclei. Characterization of the molecular basis of these processes is then possible by immunodepletion of extracts and by protein addition.
Hi, my name is Maureen, and I'm a graduate student in Dr. Maureen Power's lab in the Department of Cell Biology at Emory University in Atlanta, Georgia. Today we will show you how to form nuclei in vitro using fractionated Xenopus egg extracts. This procedure involves the following steps: performing the nuclear assembly reaction and assessing nuclei formation. So let's get started.
To begin nuclear assembly, take an aliquot of fractionated cytosol and add phosphocreatine from a 0.2 molar stock to a final concentration of 20 millimolar. Then, add creatine kinase from a 5 mg per milliliter stock to a final concentration of 50 micrograms per mil. Finally, add 20 micromolar ATP using a 0.2 molar stock. Next, add light membranes to the cytosol at a 1:10 dilution. This approximates the amount of membrane